Welcome to the Scientists Answer Your Questions set of webinars presented by GrassrootsHealth. I wanted to bring your attention this morning to the fact that these free webinars are sponsored by the participants of the D-Action projects of GrassrootsHealth. We encourage any of you that aren't already participants in that to join today to help sponsor the seminars and certainly learn more about your own vitamin D status and health. Today, it gives me absolutely great pleasure to introduce one of my delightful scientist friends here, Dr. JoEllen Welsh from the State University of New York in the Center for Excellence in Cancer Genomics. What Dr. Welsh is going to be talking to us today really is, how does it work? We've heard a lot about vitamin D does work in various areas of cancer, and one of Dr. Welsh's specialties is figuring out how it works. So Dr. Welsh, it's all yours.
Great, thank you, Carol. As you probably know in the audience, we do have a lot of information connecting vitamin D to breast cancer prevention and treatment. We have epidemiological data and intervention studies that have shown that vitamin D can reduce the risk for breast cancer. We also have a couple of studies that have shown that vitamin D status can predict better survival if you are diagnosed with breast cancer. And we also know that a good majority of patients with breast cancer are vitamin D deficient. So there's still a lot we need to know about how vitamin D works in cancer. And so today's focus is going to be on these basic mechanisms of breast cancer and some of the laboratory studies we have done to address the effects of vitamin D. So cancer is a very heterogeneous and complicated disease. There are many different subtypes of cancer, and in particular with breast cancer, there are specific molecular subtypes that have been identified, as well as heterogeneity in the clinic in terms of the prognosis and the treatment for various types of breast cancer. But when we look in general at cancer cells, we can identify various hallmarks of these cancer cells and how they behave. Things like high rate of proliferation, resistance to cell death and negative growth suppressors, and high rates of angiogenesis, high rates of immortality, genomic mutations, invasion, metastasis, inflammation. And what's interesting is in the laboratory, studies have shown that vitamin D actually impacts on all of these behaviors that we know define cancer cells and their aggressiveness. So most of the studies that have been done in the past have taken cancer cells into the lab and treating them with the active form of vitamin D, the 125 dihydroxyvitamin D or calcitriol. And these types of studies have shown that vitamin D can regulate all these processes. Now, what we've been studying in my lab is the mechanisms by how vitamin D does the inhibition of these cancer cell behaviors. One of the things that we absolutely know is that the active form of vitamin D binds to a vitamin D receptor, which is inside the cells. Now, typically, cells have this vitamin D receptor. In this little schematic here, you can see the blue ribbon diagram is the vitamin D receptor, and right there inside the binding pocket, you see the vitamin D molecule. So this complex is what basically does all of the important functions that we attribute to vitamin D. Now, what's important to know about this receptor is that it's present in all normal tissue. And by studying the actions of vitamin D binding to this receptor, we know that this complex regulates hundreds of processes within cells. And what's really interesting is that in different cells, vitamin D through this receptor does different things. There are different effects. So there's enormous complexity to what vitamin D can do inside cells. We know that this receptor is also expressed very highly in mature breast cells. So in this little picture here, we see a breast duct, and the brown staining identifies the cells that have high levels of the vitamin D receptor. And you can see there's a lot of brown here. So good expression of vitamin D receptor in normal breast tissue. Now here is a breast cancer. What you're looking at here is, as I mentioned, cancers are heterogeneous. They're made up of cancer cells, but other cells like immune cells and fibroblasts and blood cells and lots of different cell types. Now we see here that these are the actual cancer cells that are stained brown, meaning they have the vitamin D receptor. About 80% of breast cancers show expression of this receptor. So that means that potentially these cancers could be affected by vitamin D in the system. Now, we've been interested in first finding out what vitamin D does in normal breast cells. One of the ways we do this is that we get cells which are isolated from surgical resection tissue, so normal breast tissue, isolated and grown in the lab. And when we look at these cells, we see that they too have this good expression of the vitamin D receptor. They also have expression of the 1-alpha hydroxylase enzyme. Now, this is the enzyme that is able to activate vitamin D to the active form 1,25-dihydroxyvitamin D that binds to that receptor. Now, one of the things that puzzles us in the vitamin D research area is how does vitamin D get to the cells? We know that we have vitamin D in our blood in the form of the 25-hydroxyvitamin D, but we know that it's bound oftentimes to the D-binding protein. Well, we took these breast cells and we incubated them in the lab with 25 vitamin D bound to the D-binding protein, which was fluorescently labeled so we could follow it. And what we showed is that the normal breast cells just take up that 25D DBP compound or complex very readily. And you can see here the green staining depicts that D-binding protein getting into the cells. Now, when the cells take up the D-binding protein with the vitamin D, we're able to show that they could very rapidly, within a few hours, begin to convert that 25D to the 1,25-dihydroxy-D, which is the ligand for the vitamin D receptor. So we see very good expression of this, this metabolic enzyme and very good function of this enzyme to produce the active form of vitamin D in these cells. So this means that breast cells can actually utilize the vitamin D in the form of the 25-hydroxy-D in our blood to make 1,25D to perform the vitamin D actions in the cells. Now, the actions that we're talking about here is when we incubate these cells with 25D, we see that that reduces their proliferation. And it induces them to become what we call quiescent, which is basically quiet, differentiated, function, performing the normal functions of the human mature breast cells. And so that's what you're seeing here is when we use physiological concentrations of 25D, as you would find in the blood of sufficient versus deficient individuals, we see this dose-dependent reduction in the growth of the cells. Now, we believe that's a good thing because we want cells in a mature tissue to be quiescent. They shouldn't be proliferating because that's a risk factor for cancer. So these physiological relevant concentrations of 25D are able to induce this quiescence and differentiation of these normal human breast cells. When we look at what the cells look like morphologically, we can see in the control cells that are not treated with vitamin D that they are rapidly proliferating. And when we treat them with vitamin D, and again, the 25-hydroxy-D, they spread out, they adhere to the plate as they would in the normal tissue, and they differentiate. So this is all indicative of a anti-cancer tumor suppressor type of effect in that the cells stop growing and become differentiated. Now the question is, how do these changes reduce the risk of that cell becoming cancerous? There's many, many ways in which vitamin D or any other tumor preventive agent could prevent this risk of becoming cancerous. I'm going to give you 3 examples. The first example is that we have shown in our lab that when the cells are exposed to vitamin D, that protects them from DNA damage. And DNA damage in the form of mutations is the major risk factor for cancer. So one of the very interesting things we found is that when we expose cells, these normal breast cells, to drugs that damage the DNA, we saw that they immediately upregulate the amount of the vitamin D receptor in the cells. So what you're looking at here are a plate of cells, and you can see everywhere there's blue, there is a cell that are not exposed to a DNA damaging agent called doxorubicin, and those that are exposed on the bottom panels. And here we've stained for the vitamin D receptor, in this case with green dye. So when you see in the control cells, they have a low level of vitamin D receptor as long as they're healthy. But if you expose them to this DNA damaging agent, you see a very massive and very rapid upregulation of the vitamin D receptor, as if that's a good thing in the way of the cell trying to protect itself against that damage. And in fact, if we then incubate the cells with vitamin D before we expose them to the DNA damaging agent, we see that vitamin D protects from damage. Now, this may be a little bit confusing because here again we're using a green stain, but in this case the green stain is measuring the damaged nuclei, the damaged DNA. So here you see in these cells, in the control situation without any vitamin D, when they're exposed to damage, most of these cells do experience DNA damage and stain green. When they're pre-treated with vitamin D, this damage either doesn't happen or it's repaired very quickly. We haven't figured out exactly the mechanism yet, but clearly there is less damage when the cells are exposed to vitamin D, and therefore we believe that this is a protective effect to reduce or minimize the amount of DNA damage that persists under these conditions. So what's interesting is that these effects of 25-hydroxy-D and the VDR are very similar to the effects of P53, which is a very well-characterized, well-known tumor suppressor gene. So this gives us a really important clue that vitamin D does act as a tumor suppressor. The second example I'll give you is in terms of vitamin D regulating the immune response in breast cells. Now, many of you probably have heard about vitamin D regulating the immune system by acting on macrophages and monocytes, which are immune cells. Here we're talking about vitamin D regulating cytokines and other immune proteins in breast cells. Now, this is a really new finding that hasn't really been explored. But we did genomic screening and proteomic screening of these cells after treating them with vitamin D, and we found that many of the changes that vitamin D induces have to do with the immune system. And so here's one example, which is a CD14 cytokine. This is an anti-inflammatory cytokine. And what we show is that when we expose the cells to 25D, they begin secreting this cytokine like crazy. And you can see it here on the graph where we measured it with a special assay, and you can even see it here where we've stained this CD14 cytokine with a green stain. And you can see almost every cell is producing huge amounts of this cytokine when they're exposed to 25D. Two other cytokines that we found to be regulated by vitamin D in these breast cells are interleukin-2 and interleukin-15. These are proteins that, when secreted by the breast cells, we think that it activates natural killer cells, which are the cells that survey for damaged cells and eliminate them so that they cannot become cancerous. So we think that these effects are protective in that they activate the immune system and also suppress inflammation. And we know that inflammation is linked to cancer. Chronic inflammation does stimulate damage and mutations within normal cells and can increase the risk for cancer. So we think that vitamin D actually works in two different ways here. It's suppressing inflammation, but at the same time, it's activating the immune surveillance mechanisms that can destroy damaged or precancerous cells. So the bottom line on this part is that vitamin D can regulate many anti-inflammatory proteins in the normal breast cells. Now, the third example is that we have evidence that vitamin D reprograms cellular metabolism. Now, why is this important? Because cancer cells require a shift in their energy metabolism to keep them very rapidly proliferating. And this shift has been well characterized. It's called the metabolic shift, and it's known to drive cancer progression. And what we found when we did our genomic screening is that vitamin D regulates a lot of metabolic genes in these cells. And two examples are shown here. It regulates amino acid transporters that are involved in taking up amino acids from the external microenvironment into the cell. And it also regulates enzymes that control energy metabolism. In this case, I'm showing you glutamine. And so these changes actually shunt, that are induced by vitamin D, they shunt these amino acids and carbohydrates in the cell towards differentiation and towards protection against oxidative stress, rather than using these nutrients for proliferation. So the bottom line here is that cancer cells have to adjust their metabolism of amino acids, of carbohydrates, sugars, and that's needed to support the rapid cell proliferation associated with cancers. And what vitamin D does is prevent this metabolic switch. So I just summarize at this point of in normal cells, what does vitamin D do? It maintains the differentiated phenotype, and that protects against cancer via multiple mechanisms. So we know that we consume or we generate vitamin D. It's converted to 25D, and that 25D can be taken up by the normal cells. Now, I've shown you breast, but similar things have been shown in colon, skin, and prostate cancer. We also know that we have 125D that can come into the cell. And these induce differentiation, and then the differentiation is involved in preventing cancer and perhaps slowing the progression of early stage cancers. And it does this through multiple mechanisms involving metabolism, DNA repair, cell stresses, protection, anti-inflammatory effects, as well as angiogenesis, which I did not show you. So that's in normal cells. Now, one of the things we've done is compare the effects of vitamin D in normal cells and in cancerous cells, and we see quite dramatic differences. Now, this is a complicated graph, but basically it's showing you a gene profile, and each of these symbols refers to a specific gene, and the top 20 genes are shown here. And the fold change in that gene when it's treated when the cells have been treated with vitamin D. And you see both the normal and the cancer cells have high levels of CYP24, which is a known vitamin D target gene. But the normal cells shown in green have much higher gene expression changes in response to vitamin D than the cancer cells. And so we think that the cancer cells somewhat lose sensitivity to vitamin D. So if we look at normal cells, there's about 200 genes that are changed after treatment with vitamin D, whereas in cancer cells, there's only about 20 genes. And what's really interesting is if we compare the gene list, we see that in normal cells and in cancer cells, different genes are changed by vitamin D. There's only 3 genes that are common. And this means that vitamin D and the VDR have different effects to prevent cancer than to treat cancer. The effects in normal cells are different than the effects in cancer cells, and that there's distinct mechanisms that we have to better understand about what vitamin D is doing in each cell type. We have done studies, as I mentioned, by treating vitamin D onto breast cancer cells. And instead of differentiation, what we see is actually apoptosis, or cell death. So these are human breast cancer cells. These are control, and these are vitamin D treated. Again, this is treatment with 125. And these cells here are actually dead cells. You can see they're lighting up with a cell death marker. And these other cells here are on their way to early stages of the cell death process. So vitamin D can induce cell death in breast cancer cells, which it doesn't do in the normal cells. And we showed by using cells from vitamin D receptor knockout mice that the effect of vitamin D is the same in mouse cells, but that it requires the vitamin D receptor. And if we have cells from the VDR knockout mice, they do not have this response to vitamin D. But we can restore this by introducing the vitamin D receptor into these cells. So the last thing I'll show you is studies we're starting to do now in freshly isolated breast cancer tissue. So we have taken human breast cancers obtained at biopsy and isolated them, incubated them in the lab with vitamin D, and done this gene profiling. We show that these cells these tissues express the vitamin D receptor and retain their viability. And we have done genomic screening on these cells. And what's interesting is this is data from 5 separate patients. Each patient tissue responds differently to vitamin D. But we can, again, with all of these genes, we can identify a common set of genes that are changed when all of the samples are treated with vitamin D. So we identified about 50 new vitamin D receptor target genes in these experiments. And we're starting to show that, to actually explore which of the are the important targets and what are they doing to mediate tumor suppressor effects of vitamin D in these tissues. So, I mean, basically these studies show that human tumors are sensitive to vitamin D, but maybe the active form or higher doses might be needed to induce the anti-cancer responses. So to sum up, this is the model that we have. We believe that breast cells, normal breast cells, and breast cancer cells express the vitamin D receptor. Sometimes they express the 1-alpha hydroxylase enzyme. They can internalize the serum vitamin D bound to the D binding protein through a specific set of proteins on the membrane. It's also possible that some of the 25D can directly enter the cells in the absence of this importation mechanism. We also have 125D in the serum. So within the breast cell, we believe there are pools of these vitamin D metabolites that can activate the vitamin D receptor and then reduce the risk of that normal cell becoming cancerous. And it may increase apoptosis and have anti-cancer effects in cells that are already cancerous. Okay, that's it.
Dr. Welsh, thank you very, very much. Our group has a number of questions for you, some of which we will answer. But first, I want to tackle the most common question we get at almost all of our presentations from our own research study at GrassrootsHealth. The question is, how much should I take? And the very blunt answer is, we don't know. And the we don't know is because everybody's metabolism of vitamin D at this point is really quite different, and it depends on so many different factors. The best we can do is to give you a guide. Notice the green bars, the vertical bars there between 40 and 60 nanograms per milliliter. That is the scientist panel recommendation of where your serum level ought to be based on known evidence about what's good for you and the research that we have. And here's an example where that green arrow parts to, if you are starting at about 20 nanograms per milliliter and you want to get to 50 nanograms per milliliter in that target range, you can start out by trying about 4,000 international units a day. You have to measure test your vitamin D after about 3 months to see, did you get there? Because we really can't tell what's going to happen with your body in your climate, with your metabolism without testing. So that's a quick chart for you. It's on our website, and you can take a look at that. Right now, we're going to proceed with some other questions of Dr. Welsh, and let's get started. Number one, what is the difference between breast cancers that express the vitamin D receptor, the VDR, and those that don't?
Well, that's a good question. We don't really know. There are lab studies that show that when cells become cancerous, they tend to lose expression of the vitamin D receptor. But we do know that tumors do have VDR. I showed you that, and we know that from the clinic. And there are some studies out there that show that tumors that don't have vitamin D receptor are more aggressive and behave worse, so to speak. They have worse progression, worse prognosis than tumors that have VDR. But again, those studies are small and they are not really conclusive. There is not a routine test for the tumor vitamin D receptor in the clinic as there is, for example, for the estrogen receptor in breast cancer. Maybe that's something we should do. At this point, that is not routinely done.
All right. In the 80% of breast cancers that express the vitamin D receptor, does the VDR function normally, or is the receptor's function inhibited?
Again, we don't know. There is no test for us that we can do to show whether the vitamin D receptor in a tumor is functioning. As I mentioned, we're trying to identify specific targets that the vitamin D, through the vitamin D receptor, turns on or off in tumors that we could use to possibly get an idea of whether that receptor in the tumor is functioning or not. But we don't have such a surrogate test right now. We know the vitamin D receptor protein is there, so that's a good start. But we don't know in any individual tumor whether it's functioning. I showed you, though, that in the breast cancer cells and in the tissue explants that we made fresh from human tumors, they all responded to vitamin D. So they all must have some level of vitamin D receptor function. Is it top level? Is it optimized? We don't know.
It's amazing how many things that depend upon our ability to measure them in order to answer these questions.
Yes,
and we just
we
don't
really
have all of
need
those
to
yet.
do more basic research to get,
Right.
those surrogate markers.
Exactly. Question 3, does vitamin D have an equal role in breast cancer prevention and treatment, or is it more effective at one over the other?
Well, as we just talked about, because the vitamin D receptor may be less active, at least in some cancers, as they progress, we believe that vitamin D is probably more effective at preventing cancer. And we're focusing on that in some of our research studies, as I mentioned. But of course, there may be ways to optimize vitamin D receptor signaling in tumors that have the protein there. So one of the other approaches would be to try to identify ways to boost vitamin D receptor level or to activate that receptor in tumors that are already existing. So I think there's a role for both prevention and treatment, but if I had to say where is it more effective based on what we know now, I would say prevention.
All right. Last question on this page. A recent epidemiologic study found that women who had deficient serum vitamin D levels in the 3 months before a diagnosis of breast cancer had 3 times the risk of breast cancer compared to women in the highest serum level. Are there any animal or biological mechanism studies to support this finding, or have any other life periods been identified where sufficient vitamin D is critical for the development of breast cancer?
Okay, again, this has not really been well studied. We know for breast cancer that there are critical periods. There have been studies for breast cancer in general to show that, manipulations even in utero, during adolescence, during pregnancy, that these have big impacts on future breast cancer development. Now, there's not been all that many studies. I know there's one good study where they looked at sun exposure as a surrogate vitamin D marker throughout the lifetime, and they showed that high exposure to sun during adolescence, during young adult, during even in as late as menopause, all were protective against breast cancer. So that's a good thing to show that, even boosting your vitamin D level late in life is still going to have some protective effect. But again, we need more studies. Most of the animal studies have been done with young animals, growing animals, which more model, a young adult, not a menopausal age woman where most breast cancer develops. So again, there's more research that we need to do on that topic.
All right. For those of you that need to depart at this point in time at 10:30, we will give you our final slide here, and then Dr. Welsh has agreed to stay on to answer some more questions. So if you have time to stay on for that, please do so. But next week we will have Dr. Hollick talking about vitamin D is linked to gene expression. Again, it starts at 10, and he will be doing a presentation followed by questions. We encourage you to help sponsor these webinars by joining D Action today, taking care of your health and your vitamin D levels, and in, obviously, moving research into practice. So I am going to resume the question and answer session back with question number 5. Dr. Welsh, what role do vitamin D levels play in the recurrence of breast cancer?
As far as I know, there's been one really nice study by Dr. Goodwin in Toronto, which showed that individuals, women that had higher serum 25D levels, were less likely to have their breast cancer recur than women that had low levels. So that was a really nice study that did show that vitamin D could affect recurrence of breast cancer. There is some other epi data that has been done, and there is some NHANES data, the National Health and Nutrition Examination Study data, that did link mortality from breast cancer with serum 25D levels. So there is some data out there to suggest that vitamin D can be protective in women that are living with breast cancer to reduce the risk of recurrence.
I'd like to highlight on number 5 about the recurrence, the Goodwin study. One of the things that captured my attention was the fact that the reduction in risk was fully 50% if the serum level at the time of diagnosis was only 30 nanograms per milliliter. So those women who had that higher level versus a less than were 50% less likely to get to have a recurrence. As many, I think it was 12 years later, if I'm not mistaken. So
yeah, that was a really nice study. There was a clear dose-dependent effect too.
Yes. Number 6, vitamin D shown to benefit patients with more serious forms of breast cancer, such as triple-negative tumors, which are more resistant to treatment.
So with breast cancer, as you just pointed out, there are many subtypes, and the triple-negative or basal-like breast cancer is the most aggressive and the least treatable. And there really hasn't been much in the way of studies to actually compare the sensitivity or the actions of vitamin D in the different subtypes. Now, one of the problems is that some of the subtypes are quite rare, or they occur at less frequency. So in the epi studies, oftentimes they're not large enough to stratify by subtype, and this is rarely done. This is one of the weaknesses of our data sets right now. There were 2 recent studies that, within the last couple of years, that showed, though, that women with triple-negative breast cancer were the most likely to be vitamin D deficient and to have the lowest vitamin D levels. So boosting their vitamin D levels is certainly not going to hurt and could potentially help. Now, this needs to be studied both at the basic level and at the observational level. And as I said, this really hasn't been done very well.
question number 7, why might vitamin D's action be different in one type of cancer versus another?
So as I mentioned, each cancer is unique, and it has different genetic mutations that affect the growth, that affect how it responds to nutrients and drugs. In normal breast cells, as I showed you, you might have 200 genes altered by vitamin D. Now, in a cancer cell, some of those genes or pathways might be mutated, and so they can't be changed by vitamin D anymore. They might not be functioning. Some of the pathways that vitamin D regulates, like cell death, may have been altered in that cancer cell, and so vitamin D can no longer trigger apoptosis if that cell is resistant to apoptosis because of upregulation of various anti-apoptotic genes. So clearly, the mutations that underlie the breast cancers will really determine what vitamin D can do. Now, the other thing is that some breast cancers have unique signature mutations, like we have the HER2 new positive breast cancers that have an upregulation of that growth factor pathway. So that could affect the sensitivity to any tumor suppressor like vitamin D. So it all comes down to the molecular changes inherent in that particular cancer, which is going to be individual for any specific cancer. And we really need personalized medicine to identify an individual's tumor and what the molecular makeup of is, so we know what vitamin D can be predicted to do.
What do you see as the next steps in research regarding vitamin D and breast cancer for the cellular, molecular, and animal studies?
I think that one of the biggest gaps that we have in our knowledge is how much vitamin D is actually getting into breast tissue, into breast cells, into breast cancer cells. So very little is known about the uptake and the utilization of 25D by the breast tissue. So we really don't know, how much 125D is present at the tissue or the tumor level, and whether vitamin D accumulates in the tissue sufficiently to optimize vitamin D receptor signaling. And therefore, if we had info on that, we'd be able to predict what blood levels do we need to achieve to saturate that tissue with vitamin D. We really need, don't you agree that's a huge gap?
I agree. Sure.
And then from there, we would be able to predict how much supplement or diet or sunlight a person needs.
Without doubt.
I think that is something that's very hard to measure. We really can't detect the sort of minute amounts of 125D and 25D in the actual cancer cells. We need new technologies to do that.
And certainly from our point of view at this time, even not knowing that, because so many people are truly deficient, getting one's serum level up to that 40 to 60 nanogram per milliliter is a very well-advised step. Yes, crucial. Number 9, what are the main concerns when looking at animal studies versus human studies? How reliable and useful is the information gathered from animal studies for human physiology?
Well, with respect to breast cancer, we do have specific animal models. I didn't talk about any of our animal stuff, but we do have animal models that mimic all the different subtypes of breast cancer and the different stages of the disease. So we can pick one animal model if we want to study metastasis. We can pick another animal model if we want to study the HER2 new positive type of disease. So this is well worked out at the cell and molecular level. And that way, animal studies can be really insightful because we can do lifelong studies, we can precisely control the vitamin D intake and blood levels, and we can even manipulate the genes that regulate the vitamin D pathway, the CYP and the hydroxylase enzymes and the vitamin D receptor, and see what happens to the cancer process. With respect to vitamin D, we also know that most of the vitamin D signaling is conserved between human and mouse. So we have mouse models where we have VDR knockout or we have the hydroxylase knockouts, and these very closely mimic people with mutations in these same genes. So we've also shown, and I think I had this in one of the slides, very similar effects of vitamin D on mouse cells and mouse breast cancer cells as we see in the human normal cells and cancer cells. So there's one caveat, and that is the immune system. We do see differences in some of the genes that are regulated by vitamin D that mediate the immune responses in mouse versus human. But basically, we feel that most of what we find out in mice with respect to vitamin D and with respect to cancer can be extrapolated to humans. The hardest part is the extrapolation, because when we do a mouse study or a cell study in the lab, we have a very homogeneous system. So not because of species differences, but because the mouse models, we can be very well controlled. And in human patients and tissues, we have extreme heterogeneity. Each cancer that develops in an individual is unique at the molecular level. It may be called breast cancer, but that's because it develops in the breast, not because it's similar to another breast cancer necessarily. And so only by studying large numbers of cancers can we come up with generalizations, but the application of those generalizations to any individual patient is tricky. So again, we need this personalized medicine so we can really get a handle on this at the individual level. But basically, I think the animal studies and the lab studies have been extremely informative for getting at this molecular information and finding out the vitamin D targets in tissues.
Question number 10, does vitamin D protect against damage from toxicity or oxidative stress as a method of decreasing cancer incidence? What about protecting against environmental toxins that have been tied to breast cancer, such as parabens? If so, how does it work to do this?
So as I showed you, we know that vitamin D protects against DNA damage, which is a major mechanism by which a lot of these environmental factors increase cancer risk. There's other studies by a couple of other labs that have done similar studies, similar types of experiments, and shown that vitamin D does protect against oxidative stress. With respect to the parabens, I don't think they have been specifically studied with respect to vitamin D. From what I know about these compounds, though, they are thought to act as weak estrogens. We know that vitamin D inhibits estrogen signaling and metabolism in breast tissue, so it could be protective against those in that fashion. But the whole issue with estrogens is quite complicated, because we have some estrogens which are protective. The phytoestrogens, the soy estrogens, are thought to be protective against breast cancer. Other estrogens are xenoestrogens, which actually increase the risk. So to really tease that out, specific experiments would have to be done to see how vitamin D interacts with paraben actions or metabolism. I'm not aware that those studies have been done yet.
Okay. We have some more questions, Dr. Welsh.
Oh, that's fine.
This is our last page of questions, however. Is there a connection between vitamin D, estrogen, and other hormones and their effect on breast cancer? And you certainly talked about that a little bit, but I'll let you expand on it.
Right. So vitamin D does inhibit estrogen signaling. It inhibits the aromatase enzyme, which actually metabolizes estrogen. And aromatase is a major target of drugs, the aromatase inhibitors for breast cancer. So vitamin D does act like them to inhibit vitamin D estrogen production. This is important because in postmenopausal women, their tumors may be estrogen dependent, and that estrogen is coming from the adipose tissue, which has aromatase and can activate estrogen and synthesize estrogen. And so vitamin D can inhibit that aromatase, which would presumably be one way in which vitamin D is protective against breast cancer, especially the estrogen-dependent type. But we know that vitamin D does inhibit cancers that have nothing to do with estrogen, prostate cancer, for example, or colon cancer, and that the vitamin D's mechanism of action is far beyond just inhibiting estrogen action. But yes, vitamin D does inhibit many of the estrogen-dependent actions in breast cancer.
Okay. Number 12, what types of breast cancer are most responsive to vitamin D therapy? and the least responsive?
Yeah, as I said, that it's not really been well studied in relation to the molecular subtypes of breast cancer with respect to how vitamin D might selectively affect one or the other. In general, we do think that early stage cancers, as I mentioned, are more likely to have abundant VDR and intact vitamin D signaling. So theoretically, these would be more responsive to prevailing vitamin D levels in the blood. But, I hope I pointed out that each cancer is unique, and it really depends on the vitamin D pathway integrity as well as other pathways that vitamin D regulates, and many of these are often deregulating cancers. So, you could have late-stage cancers that could still be fairly responsive to vitamin D. There's no generalization here with respect to subtype, though, that I know has been well worked out yet.
Number 13, in my own practice and studies, I've noticed a connection between early menstruation and puberty and increased incidence of breast cancer. Do you see this as well? Is vitamin D involved in this connection?
So this has not really been well studied either. It is known that obesity is one of the factors that scientists think links earlier onset of puberty and higher risk of breast cancer. It's very prevalent in today's society. We know that vitamin D has impacts on obesity. It can affect adipose tissue. It can affect metabolism, as I've shown you, which may play into this. It can affect the metabolic syndrome. So we think that increasing vitamin D status would have beneficial effects on obesity and on insulin signaling within tissues, probably also the breast, since there's so much adipose tissue in the breast. And so if there is a link, it's probably through obesity and metabolism. But again, we haven't worked that out yet, the basic level.
Okay. On number 14 in your paper, you state that during puberty and pregnancy, we see increased proliferation and branching of cells. What changes during these times to contribute to this, and does this indicate a greater need for vitamin D during these times?
In the work that we've done, we have looked at the effects of vitamin D during all phases of a woman's reproductive cycle. So during puberty, during pregnancy, lactation, involution, and we do note that vitamin D has actions at each stage. And when we look for the genes that are changing at each stage, they're different. So vitamin D does specific things at puberty and specific things at pregnancy. Again, this is in the mouse model. And essentially what we think is happening is that all of these changes in the gland during these times are driven by estrogen and progesterone and prolactin. These are the reproductive hormones that are acting to stimulate growth of the gland at these periods. And we believe vitamin D is there to counteract that proliferation, to just make sure it doesn't get out of control. And it's not the only factor that is counteracting the estrogen and progesterone hormones, but it's one of the main factors. And so think of it as a brake on these very, very strong hormones that drive proliferation and branching in the tissue. And so we think, obviously, that optimizing the vitamin D status at any and all of those stages is really important to maintain that strict control, the checks and balances on, the turnover of those cells during these critical periods. So yes, we believe that vitamin D is really important during those critical times. But as I answered for an earlier question, we really haven't got the data to show in humans whether vitamin D and how much vitamin D at all of these periods is necessary.
Mm-hmm. Question number 15, how can the benefit be measured? In other words, if I had breast cancer, how would I know the cells had stopped growing by having a higher vitamin D level?
You wouldn't. There's no way. There's no way. Basically, what this would require would be a biopsy before taking vitamin D and after. And really, see, the thing is, if somebody has breast cancer, then the cancer is removed. And then you go on a vitamin D regimen, let's say. The cancer is gone. You're hoping it doesn't come back. I mean, that's your way of knowing, is if your cancer stays in remission, then, you know, vitamin D is doing its job.
Actually, it just dawned on me that we had a presentation by Dr. Bruce Hollis about 2 weeks ago about a prostate cancer study they did with a group of men who were in a stage of watchful waiting.
Right.
And by 4,000 IU a day over the year's time, they actually had biopsies at the beginning, and at the end they had fewer positive core biopsies. So those men were subjected to multiple biopsies, unfortunately.
Yes, but we don't have watchful waiting for breast cancer in the same way as there is for prostate cancer.
Maybe there should be.
Well, one of the issues with breast cancer is GCIS. So in GCIS, basically women are diagnosed with GCIS and have it removed, and then they have to be subjected to, and the vast majority of women are subjected to, radiation and tamoxifen therapy. And the vast majority of those women don't need it, but we don't know which ones need it and which ones don't need it. So, you know, you have the choice
is
at that point.
Ductal carcinoma in situ.
Right. So, you know, at that point, if that was me, I would optimize my vitamin D at that point. But to be able to measure that, the tumor is gone at that point. So, I mean, I really don't know how we could design a study like that ethically.
Well, there's watchful waiting. Number 16, is there data on vitamin D in breast cancer stem cells?
Not really. We have some data that we haven't published yet. We're starting to look at this. We have identified some targets of vitamin D pathway that seem to be critically important for the survival of these breast cancer stem cells. So we are actively looking at that. There was one study recently which looked at vitamin D receptor in mammospheres. Mammospheres is when you take breast cells and grow them as three-dimensional structures, and this function is mainly breast cancer stem cells that can grow like this. And sorry, not breast cancer, but just stem cells in general. And in that paper, they showed that the stem cells did not express the VDR. They basically lost expression of the vitamin D receptor. But I don't know if that really reflects what you see in cancers. I mean, we have shown that vitamin D is able to affect these very advanced breast cancer cells that have stem cell markers. So we think that there's some more research to do there, and that vitamin D does affect breast cancer stem cells in a beneficial way. But again, there's really not any published data on that yet.
Our question number 17, is there any progress on vitamin D and reversing tamoxifen resistance?
I'm not really quite sure what this question is asking. There are studies out there that show that breast cancer cells that are tamoxifen resistant are still responsive to vitamin D. We've shown that in vitro. We've shown that in animal studies. There are studies showing that if you combine vitamin D and tamoxifen together, then you usually get a more beneficial effect than either agent alone. But in terms of reversing tamoxifen resistant, I think this is asking, well, if a tumor is tamoxifen resistant, can it be treated with vitamin D and then become sensitive to tamoxifen? I don't think that would happen, because vitamin D inhibits the estrogen receptor, and therefore that would tend to eliminate the tamoxifen sensitive cells on its own. So I think that is an interesting area, and I think that there's possibly interactions between vitamin D and tamoxifen that could be, exploited in the clinic, but In terms of reversing tamoxifen resistance, I do not think that we have data on that.
There is so much that is coming forth, and in your studies and others, on the benefits of vitamin D and being at higher serum levels in that 40 to 60 range, that no matter what data we have or don't have at the moment, that is an advisable thing to do. On this next slide here, Carol Baggerly, am going to answer this from our research study that we've been working on. We had a lot of questions about dosage and serum levels, and I'm actually going to take questions number 1 through 4 on this page and answer them all as a group. It talks about what level would be considered toxic or not optimum, what levels are therapeutic, what's the best way to monitor it, how should I get my vitamin D levels checked, and is there a difference in the level of protection with rising levels of vitamin D? All of those go together in terms of our research project and the way our scientists have designed it. In the panel of scientists, of which we are absolutely delighted to have Dr. Welch and every one of the other people that you've heard in the webinar, the current scientific data says that you really should be at a physiological level, there's nothing abnormal about it, in the 40 to 60 nanograms per milliliter range. And that's 100 to 150 nanomoles per liter for those of you that use that unit of measure. And you take, if you're taking supplements, whatever you need in order to get there. The best way to monitor your vitamin D, quite simply, is with a vitamin D test. There's no other way. The intake, as you will see if you look at some of the charts on our website, does not determine your serum level. People that are taking 2,000 international units a day may have a serum level of 20 nanograms per milliliter. They may have a serum level of 120 nanograms per milliliter. We flat don't know what your serum level is until you test. And I, having been a breast cancer patient, and I see the significance of vitamin D, bar none, for what I should do to prevent a recurrence of it, there is no way that I am not going to test to make sure my serum level is always in that range. And the serum levels don't vary considerably once people stabilize, but regardless, it's important to check. In terms of when to check, your lowest serum level is probably right now in the March-April timeframe, right after winter. It's not a bad time to check right now, because this is probably the lowest you're going to be due to the latitude where you are, so forth and so on. So check it now and make sure you're still in that range. If not, make sure you supplement adequately to get there. And the African American person, it takes more vitamin D for you in general than it does a Caucasian person, but certainly the out of doors area. If you're depending upon sunshine to get it there, it takes longer out in the sun due to the protective coloring of the melanocytes. Is there a difference in the level of protection? I want to address that somewhat with different diseases. On one of our charts that we have, again from different research, the disease incidence prevention chart, different diseases and very likely different kinds of cancer could be considered different diseases, have different levels at which they are sensitive. If you look on the chart, you see that you can totally prevent rickets at about 20 nanograms per milliliter. That's beautiful, but you need to have it be at least at 30 to do something with bone health. The Lappy study out of Creighton University in 2007 showed by getting just from 30 to 40 nanograms per milliliter, they prevented about 77% of all cancers. And then there are other diseases in that 40 to 60 range. So again, stick for that range, and I would actually suggest you not spend too much time worrying about different diseases. Just go for that range and you'll take care of as much as you can. The last question on that page, how can this information be disseminated to oncologists, ACOG members, and family practice physicians throughout the U.S.? Dear participants, listeners, it's up to you. It's up to us. Our disease incidence prevention chart has literally been downloaded thousands and thousands of times for the express purpose of someone paying attention to our site, taking it, to their physician. We then will have a call from the physician wanting to know what's the research, etc., which we are absolutely delighted to answer. At this stage of vitamin D research and our public health implementation, we are driving it through the interested and intelligent layperson who is making it happen. And I encourage you to continue with that. Thank you, everyone, for attending today. We very much appreciate it. And Dr. Welsh, thank you for an absolutely A-plus presentation on breast cancer and vitamin D. Next week, we will be again listening to Dr. Hollick talking about gene expression and vitamin D. And again, we encourage you to help sponsor these webinars, which we get very positive responses from those of you who listen, by joining the D Action Project. Have a beautiful day, and we so much appreciate all of you. Thanks again. Bye.